Hello, my name is Lynn Enquist. I'm from Princeton University in the Department of Molecular Biology. I want to talk to you today about the future of virology, or virology in the 21st century. Now, I have a quote here from either Yogi Berra, the baseball player, or Niels Bohr, the physicist. It's not clear who gave the quote, but it's tough to make predictions, particularly about the future, and that is certainly true in virology. But we can say with some certainty that the forces driving the future of virology are five. It's new technology development. There's a lot of information that's being produced. The needs of the public health needs, the public perceptions of science and what they need, and of course personal curiosity that drives the scientists. There's one take-home message that we can deliver from all of this thinking, though, is that while we can predict the short term with things that we know now, we can't imagine the breakthrough discoveries that are going to happen. And so by definition, discovery can't be predicted. But we can say, without a shadow of a doubt, that the future of virology is going to be very exciting. Now we know this from history. Virology's had a phenomenal impact on biological discovery. Tobacco mosaic virus, or TMV, was discovered more than 100 years ago. Since then, virology has impacted every aspect of biology. It's provided fundamental insights into genetics, biochemistry, structural biology, and evolutionary virology, and there's been more than 20 Nobel Prizes awarded for work that has involved virology. In fact, we can say without a shadow of a doubt that without virology, we would not have the tools of molecular biology and genetic engineering. It's very curious that virology is a very integrative subject. A successful modern virologist must know a little bit about everything. You've got to know everything that I've listed here if you're going to really understand what's going on in virology, from immunology to physics to population biology and ecology, as well as molecular biology and public health. One thing that's really clear is that virologists have a certain kind of job security, because viruses are a deep part of the planet's ecosystem. They are found everywhere that life exists. We know this because the isolation and characterization of viruses from unconventional habitats is providing new views of virus diversity, of evolution, and of function. This virus here is isolated from archaea that grow in boiling springs in Yellowstone National Park at a pH that's approaching 1. And these virus particles are stable, and they're telling us a lot about protein structure and function and how organisms can exist in this kind of environment. That being said, our ignorance about virus ecology has been remarkable. I'm just going to give a couple of examples here. If you considered our new data on the number of virus particles in the oceans, the estimate is that at least for certain kinds of viruses that have been quantitated, there's probably more than 10 to the 31st power virus particles in the oceans. That's an enormous number. It's also been calculated that with that number of virus particles and the number of organisms that these viruses can infect, we can estimate that there are more than 10 to the 23rd infections per second going on in the ocean. It's an astronomical number. And in fact, you can take water from any ocean in the world and look at it and count the number of virus particles, and you typically will find numbers of 10 to the 6th to 10 to the 8th virus particles per ml of water in the ocean. And in fact, when you look at the abundance of prokaryotes, the yellow here, the protists, the single-cell eukaryotes, and the viruses, you discover that viruses are the far more abundant microorganism, or organism per se, in the ocean. In fact, here we have a stain that stains nucleic acid, either RNA or DNA, green, and this is a sample of seawater, and you can see it's full of puncta that are green. The large ones here are single-cell organisms, but the smaller ones are viruses. The oceans of the world are full of virus particles and infected cells, and yet we know very little about what is going on. We do know that the carbon and sulfur cycles of our whole ecosystem would be altered without viruses, because at least some of the examples that are well studied, for example, the marine photosynthetic phytoplankton that exist and fix carbon through photosynthesis, in the upper meter or so of water layer of the ocean, these phytoplankton create huge blooms in the ocean, and they are subjected to infection by specific viruses that exist in the ocean, and they lyse these phytoplankton. The phytoplankton that I'm showing here are ones that make a calcium carbonate exoskeleton, and this exoskeleton is a sink for the carbon dioxide that's being fixed, and that essentially falls down to the bottom of the ocean, and that these blooms disappear after the viruses have lysed everything. And so that we know also that there is a sulfur aspect of this cycle that's also being released when these viruses infect these cells, and so that the whole carbon and sulfur cycle of the Earth would be altered without these viruses. And so that is something that is really phenomenal. There's another surprise in the ocean and in water. A few years ago, we discovered giant virus particles that lived in amoeba. These virus particles were so big that we didn't even think that they were viruses at first. Virus particles are supposed to be very small. These things turned out to be very unusual viruses, very interesting viruses. This is a picture of the Mimi virus. This is an EM transmission electron microscope image, and this is a scanning EM image back here. These particles are over 800 nanometers in diameter. They've got 1.2, 1.3 million base pairs of DNA, over 900 open reading frames. The genome of these viruses is larger than the genome of at least 25 different bacterial genomes. These viruses are not completely on their own. They're related to baculoviruses that infect insects and a variety of different kinds of pox viruses. But once these viruses were discovered, other people started looking, and we discovered even larger virus particles are out there, and these are called the megaviruses. This is an image of megavirus chileensis. It was isolated in a water sample collected off the coast of Chile. This is one that's called Pandorovirus that infects amoeba, and it's absolutely huge. And recently there was a large virus that was resurrected of the same kinds of particles. It was resurrected from animals that were frozen in the permafrost. So if you look closely at the viruses that are out there, and you look in different places, you discover an astonishing diversity of virus particles that are awaiting discovery. Just one more example, if you look at these Wasp virus particles, these are very unusual. These are virus particles of a particular kind of DNA virus that's coming from the parasitoid Glypta fumaraceae. These things consist of several nucleocapsids. That's what these structures are. There are proteins that cover up their DNA, and there's two envelopes that surround these nucleocapsids. But the amazing thing is that these are DNA viruses. Their DNA genome is not in a single piece, and it's at least in 100 pieces, and the pieces are circular double-stranded DNA. Each one of these nucleocapsids may package more than one double-stranded circular DNA molecule, and it's not even clear if each of these nucleocapsids contains the full spectrum of the more than 100 genome segments. We just don't know anything about these particular viruses. So what's next in virology? Certainly, without a doubt, there will be new technology. Every time a new technology comes on the scene, we get new vistas. We see things we never saw before. So recently, the only viruses that we could discover or identify in a complex community, like the gut flora or in seawater, were the ones that we could grow. If we couldn't propagate them, we couldn't study them. But now, through research that's evolved in the early part of the 21st century, we can do high-throughput, deep sequencing of RNA and DNA. We can essentially sequence all of the nucleic acids in a particular sample, and this sequencing of all of the genomes in a mixed population found in respiratory secretions, gastrointestinal contents, soil or water is essentially identifying viruses and new genomes that we never knew about before. It's another example of this kind of DNA technology that's really revolutionized epidemiology, the study of viral infections in the natural world. In 2001, the foot and mouth disease, FMDV, epidemic in the United Kingdom was followed by traditional methods. They took weeks to months to analyze, and we followed this epidemic, and we knew a lot about it. In 2003, a man named Joe DeRisi, invented something called the virochip on this particular slide. He has the particular sequences from over 4,000 different viral genomes on a particular chip. And using that, he was able to identify the SARS coronavirus in a few weeks. Just completely changed the way that we look at new infections that we didn't know what was causing it. And then just 9 years later, in 2012, we had another coronavirus outbreak, the human coronavirus, or the MERS coronavirus, that arose in the Middle East, was first identified in the Middle East. The sequencing of the whole genome of this particular virus only took a few days, and we understood what was going on. Sequencing technology changes the way that you can look at a particular infection and figure out what's causing it. I found this paper to be really quite interesting. This is Ann Pulvenberg in her lab. It was published 2009 in Science. She sequenced and analyzed all of the known human rhinovirus genomes. These are viruses that cause the common cold, and she showed how they were all related and how they were, how the genes were conserved. And it was really remarkable that she was able to get all of this information, put it all together, and reveal unusual and interesting evolutionary relationships among these very common human viruses. It's interesting that the technology has now put us in a place where we might consider that we might know everything we need to know about virology via sequencing and new genetic technology. We know the complete genome sequence of many viruses. We know the complete DNA sequences of many viral hosts. We can test systematically the role of every cellular gene in virus infection using RNAi technology or the new CRISPR technology that enables us to knock out one gene at a time. And so it's possible in screens using this kind of technology, and we've seen it with HIV virus, with West Nile virus, with pox viruses. It's identified hundreds of unexpected new cellular genes that are required for efficient infection. You can also go the other way by doing host genetics, human genetics in particular. We can find differences in individual genomes, individual humans, that make them more or less susceptible to viral infections. It's called whole genome association studies, and you can identify host genes that are required for infection, pathogenesis, and transmission. So these studies, these screens of different genes that are involved in viral replication, the host genes that are involved in for infection and pathogenesis and transmission are all leading to new ideas about antiviral therapies and improved viral prevention strategies. Now, in the past, identifying pathogens, that is, organisms that are causing disease, has been difficult and slow. It's difficult to grow them, it's difficult to analyze them. Assembling the data, maintaining the databases that were necessary, and providing access for analysis by a whole variety of other individuals requires advances in software and bioinformatics. And this is just exploding in biology at the moment. Because now it's possible to collect and analyze serial samples over time as patients move from being healthy to being ill. And you can follow the changes in their gene expression patterns and the pathogens, the non-pathogens that are in their system, and you can watch what's going on. Of course, this raises another really serious problem. The challenge is now going to be how we can move from correlating the presence of an agent, the presence of a new genome or agents in a disease, to proof of causation. We certainly are finding that there are more than a few variables that are involved in trying to figure this out. I'm just going to show you one example of both the power of this kind of technology and also the interesting challenges that open up. And this is a paper that was published in the Journal of Virology on pathogen discovery by sequencing the fecal virome, all of the viral genomes that are in the feces of animals. This is particularly was done in a high-density farm that was raising pigs. And what they did was they sequenced fecal samples over time. And this is a plot of two different kinds of parameters that look at sequences. It's not important what they are. But they were able to identify the genomes of these viruses that were in these fecal samples and classify them as viruses that were from vertebrates, that's the red circles, viruses that were from plants, that's the green circles, viruses that were from arthropods or insects, that's the blue circles, and viruses that were from worms, in this particular case, C. elegans, and another particular Ascaris type worm. And what was very interesting is that they discovered that there are a lot of viruses that were in the fecal samples of these swine, and that they identified some new ones, these POSA viruses that they had never seen before. But it was really interesting looking at this to make sense of what this kind of data shows you. Which ones are the pathogens? Which ones are the passers through? Which ones are the normal flora that's in these animals? And so the identification of new viruses through this deep sequencing technology brings a serious challenge. Are they pathogens, or could they have a symbiotic relationship with their host organisms? We're starting to believe that organisms such as viruses, organisms depend on microorganisms such as viruses to actually maintain good health. Maybe having viruses in you is good for you. Maybe they stimulate local and systemic immune responses that protect us from infection by more virulent microbes. Dissecting these relationships will undoubtedly yield unanticipated insights about viruses and their hosts. To do so is going to require careful epidemiology and clinical studies. It's going to be aided in part by more advances in technology that we have, being able to analyze all of these genomes and figure out what is going on. It's another example of this complicated ecology that exists in organisms that have a microflora, the microbiome, so to speak, of bacteria and also viruses. We know that the intestinal microflora is essential for our health by limiting the colonization of pathogenic bacteria. It's a very active area of research right now showing this. And we do know that when we treat with antibiotics, we change the microbiome. Both the good guys and the bad bacteria are killed. And then it's very interesting to watch what happens when they come back. But an interesting outcome, something to really think about, is that many viral infections are enteric. That infect our enteric system. Viruses like poliovirus, reovirus, and norovirus replicate in our intestines and can have pathogenic effects. So when we treat with antibiotics and we alter the bacterial microflora of our gut, do we affect these viruses? The very interesting thing is that antibiotics severely affect the capacity of viruses like poliovirus and reovirus to replicate in mice where these studies were done. And so we're just learning what these studies mean. Clearly, the dynamics of the gut ecosystem are poorly understood. It's not just poliovirus replicating in the gut, it's poliovirus replicated in the presence of the microbiome that's we need to study. So a new kind of approach to virology has been evolving over the last 10 or 15 or so years. It's called a systems approach to virology. And instead of being totally reductionist like we have been in the past, studying just one gene or one gene product at a time, we now have the capacity to study simultaneously large groups of genes, not just the genes of the virus, but also the genes of the host. And what this technology has allowed is the identification of fundamental biological networks. So these are the complicated and interconnected intracellular processes that control gene expression, organelle biosynthesis, metabolism, defenses. We know that whole networks get turned on or turned off after viral infection, for example. And understanding these networks turns out to be a critical aspect of the future of virology. So the fundamental premise of this kind of holistic virology, studying everything at once, so-called systems virology, is that information, biological information, flows through networks. And disease arises when these networks are perturbed. Infection changes network architecture, and the dynamics of information flow. And as such, we're going to be looking for viral signatures of network imbalance as opposed to just changes in one or two gene products. So we're not affecting one cellular pathway, but infections are altering the fundamental homeostatic balance, which is necessarily dynamic, of a cell, organism, or even a population. This is just an illustration of a network that's looking at a variety of different kinds of proteins and signal transduction that's evolved from the EGF receptor that binds a growth factor and turns on a whole variety of other genes that are there. After viral infection, we can expect that this whole network will change. Other new technologies are coming quickly to fill out the premise of systems virology. We have a lot of new and powerful techniques that are showing up. We can do whole genome expression studies. We can measure every transcript, every protein. every single ribosome. We can map them on host and viral genomes, and we can put ribosomes on as they're synthesizing their mRNA on different genes. We can look at polymerase pause sites, ribosome pause sites. We can study a whole variety of things about gene expression. We can look at every protein, every lipid, every metabolite by mass spectrometry. We can look at whole cell, organelle, and single molecules through imaging technology, very powerful imaging technology where we can watch an infected cell from beginning to end, and we can also watch an infection spread in living animals. So this raises a very interesting question, at least for some of us of a certain age. How can we couple new technology with established procedures? Will the methods of traditional virology continue to be necessary? Can we turn off our incubators and freeze down our cell lines and essentially just do deep sequencing and use our computers? Will the plaque assay, standard kind of way of titering viruses, go the way of the Model T, the first cars that appeared? For me, the answer is fundamentally no. Experiments with cell culture, biochemistry, animal models, clinical trials, population-based analyses, they all will be essential components of contemporary and future virology research. We have to know how to test the hypotheses that are raised by systems virology. So I believe that the virology toolbox will be enhanced by technological innovations rather than replaced by them. So the new technology and the new questions that arise are really very challenging. So a couple of them. How do we characterize new viruses discovered through deep sequencing? We only see them as genome sequences. We don't really see them as viruses that we have in our hand. How are we going to characterize these? And there's a lot of interesting technology that's going on with them. How do we investigate viral pathogenesis and disease outcomes with nothing but ensembles of genomes? How do we know what genomes are doing what? Deep sequencing is providing a lot of challenges. So causal association of viral genomes with specific disease phenotypes requires experimental infections and intervention trials where we can actually block some of these genomes from being expressed to see what goes on. There's something that's maybe not so obvious to scientists as we think about the future of virology. We absolutely depend on the public. The public has to need us. They have to feel that we're doing something important, and the public also supports us. The public provides much of our research funding. So another job of virologists, if we're going to have a good future, is that we've got to foster public support and advocacy for virology research. So scientists must make it clear that economic stability is interwoven with scientific progress. The public must understand science and scientists. Our major message has to be that as virologists, we seek to understand basic biology and ecology. This knowledge is going to lead to improvements in the health of the flora and fauna that inhabit our planet. It's not our job to essentially go out and think about finding antiviral drugs and therapeutics. That's a necessary outcome and a good outcome of what happens. But we can't do that unless we understand basic biology and ecology. So this brings me to the concept of training. How are we going to take advantage of all these new opportunities for scholarship and for application? How are we going to train virologists for the future? What constitutes optimum training for virologists in the 21st century? Well, I believe that tomorrow's advances will require an interesting combination of groups. There'll be small, tightly focused research groups, and there'll be large, multidisciplinary teams. We have to have both in order to have progress. Interdisciplinary teamwork is extremely powerful. I spent a large chunk of my career in industry, and I know how important teamwork is to move projects forward so that you can get products at the end of the line. But this is not just basic scientists working with clinicians and veterinarians to talk about human health and animal health, but it's also important to have mathematicians and physicists and population biologists working with us. We not only have to understand the animals, but we also have to understand the plants and the insects and the whole variety of workings of the ecosystem so that viruses can inform us what's going on. So the trick, of course, has to be that everyone has got to speak and understand each other's language. An orchestra makes beautiful music because everybody understands what the conductor is trying to do and they can read their particular music. But if everybody is working for themselves, you get a cacophony and not a symphony. It's a major challenge. Something that's really important to me, and I see that some people, particularly that aren't scientists, don't appreciate the real joy that scientists feel. Personal curiosity. That's what drives discovery. I mean, the power of a single mind to identify a problem and solve it is one of the greatest joys of humanity. No executive, no laboratory head, no business manager or marketing director can tell you how to discover something. Discovery has to come from being prepared and being in the right place at the right time. The very meaning of the word discovery is that we didn't know what we would find when we started looking. You can't dictate discovery. The process of discovery follows paths that seem simple or elegant in retrospect, but they almost always reflect individual intellect, personal curiosity, and luck. It just can't be defined or packaged. You have to provide the opportunity for discovery. I mean, you can see this when you look at virology discovery history. All those Nobel Prizes give us a good opportunity to look at things. Serendipity, the ability to all of a sudden see something that nobody else saw before, is an essential force in discovery. How do you harvest serendipity? Someone has to see something no one else did. They have to be in the right place at the right time, and they have to be prepared. So we can predict that discoveries in the 21st century will occur because someone wants to understand how something works, not because it's trendy or not because it's fundable. These people who are going to make the discoveries will be looking for the void in what we know. What don't we know, and why don't we know it? Instead of just trying to embellish what we already know. So let's look into the crystal ball a little bit. We can't predict the future very well, but we can certainly say, based on everything that I've seen and some examples that I've told you, that these are exciting times for biology in general and for virology in particular. The knowledge, this extremely powerful new technology, new ideas, the urgency to learn more are just stronger than ever. I can tell you what the obvious drivers of virology research are going to be for the next 10 years. They're going to be systems biology of virus-host interactions, viral ecology and the virosphere, and this is not just plant, animal, and human research. This is going to involve every living thing on the planet. It's going to tell us about evolution of viruses in the real world. Where do they come from? Where are they going? How are they getting there? We're going to have efforts to improve vaccines and find new therapeutics to improve the health of of humans and animals and plants. We're going to determine the dynamics of pathogenesis, how disease occurs. We're going to understand zoonotic infections. These are infections by viruses that are already out there and other species that will invade and infect humans. We're going to understand those things. And that means we have to understand population biology and public health. These are the 6 drivers of virology research in the next decade that I can predict with some confidence. But we are at a seminal moment in the conduct of life sciences. There are a lot of things that are going on that we have to watch. Some people have concerns about biosafety and biosecurity. These concerns have got to be addressed. The public trust must be cultivated, because remember, the taxpayers fund most of our work, and so they have to see that we're doing our research safely and conscientiously. I used to be editor-in-chief of the Journal of Virology, and I spent a lot of time during those 10 years thinking about the future of journals and traditional publications, and right now I can tell you that things are really changing. Scientific communication is changing in many ways. In the next few years, there may be no more print journals. Everything will be electronic. We're going from into a system where there's more open access, where journals are published and everybody can have access to them. We're seeing more reliance on impact factors, citation indices, to really tell us whether the work is useful or not, or can be used for promotion of people or assessment of their prowess. There's a big discussion about whether impact factors are useful or useless. Peer review is undergoing an interesting sort of analysis right now, where the traditional ways of reviewing manuscripts and by anonymous reviewers is being questioned. Certainly at a time when you can essentially publish anything that you want on the internet without any kind of peer review raises the question, again, about how we're going to communicate science. But one thing is really certain. The basic biology of viruses, even those that today may not seem relevant to human, animal, and plant disease, have got to be studied. We've got to understand the role of all viruses in our planet's ecosystem if we're going to make any progress. I like this cartoon because it shows a caveman talking to his partner. I was just rubbing sticks together for fun and I didn't realize I was doing basic research. That's the idea of serendipity. That's the idea that you can study something and then discover something that's going to have a totally different relevance. And that's something that I really value. So I'm a cautious optimist about the future of virology. I believe the future is bright. There's very exciting things going on. It really is an exciting time to be a virologist. So I thank you very much for listening to my lecture.